Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonise approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of the discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
Let's start. Welcome everyone. In case you want to know where you're at, this is the Test Driving Valid 2021 session, co-organized by Shannon Bennett, who's on the call. You see him on screen, and myself with the help of many others, among them Ulla, who will also provide sort of an update on the website. So maybe briefly, I think everyone who's here knows that we're trying to go through some of the, proposed concepts of the Valid Act, and I have a copy in front of me, and a lot of these concepts are obviously new and sort of undefined, so at some point we have to probably make some assumptions of what potential guidance from the FDA or another regulatory body would look like. But to keep things sort of like on track regarding the, let's just call it pathology focus, etc., we picked three test scenarios that I think we could possibly start with briefly, just as an outline. I know that some people emailed me that they may be a few minutes late, so let me just quickly go through this. So can you see my screen?
Yes.
Perfect. So the first let's just call it use case that we discussed was a whole slide imaging based AI application to predict the combined Gleason grade from H&E stained routine formalin fixed paraffin embedded sections. So clearly we can wordsmith this a little, but we can define later on what sort of the intended use, etc. is. The second one would be a PD-L1 immunohistochemical staining for a solid tumor, as in this case defined for a companion diagnostic application that is cross-referencing another approved test. So we can pick a specific cancer or go just in general through this. So this would be an immunohistochemical stain. And then the third use case was a next generation sequencing assay. So a probably comprehensive DNA genotyping assay where multiple established biomarkers are assessed in a comprehensive fashion using, in this case, a sequencing technology. So the idea was to cover different complexities, one more traditional, H&E based, let's call it architectural biomarker, one immunohistochemical stain in the companion diagnostic field, and then sort of a multi-analyte biomarker. So I hope that makes sense in terms of like the use cases. Shannon, did you want to add something to this, or?
Oh, I think that's good. I mean, I'm assuming that if folks are on the call and are interested, they have a baseline understanding of kind of the current state of FDA regulation of LDTs and what the VALID Act hopes to accomplish. If anyone doesn't have that background, feel free to speak up and we can give a quick synopsis of what VALID is.
So, I know some people on the call, but should we do a quick sort of like, I know Gina and Norris, you just introduced sort of who you are and what I know Steve, Jennifer, Eric, Rashni, do you want to maybe briefly provide sort of a background of what your interest and role is, just so we have sort of a context? Hey, Eric.
Sure. So, go
My name-
ahead.
Okay, yeah, my name is Rajni Mandel. I am a graduate of MGH, Path and Search Path. I'm also DermPath boarded, and I work for Pathology Watch, which is a digital pathology DermPath company who is also working into AI.
Perfect. Thank you. Good having you, Rashni. Thanks.
I'm Eric Connick. I'm an assistant professor at the University of Washington, Seattle, and then regulatory affairs is one of my perverse hobbies.
Thank you. And Jennifer on the phone?
Yes, hi, I'm Jennifer Dickey. I'm a Vice President Regulatory at PGDX, and I was at the FDA when the first round of LDT stuff was coming through, so I'm just interested in the case studies. Thanks for having me.
Perfect. Thank you for being here. So I think the key element is obviously risk mitigation. I think that's one of the big sort of overlying concepts. An IVCT definition, and then of course risk classification. If I'm reading the VALID Act correctly, and then if we would deem one of these tests low risk or high risk, then how to potentially implement some mitigating measures. I think that's sort of some of the key concepts here. So I don't know if we want to discuss this individually, or should we just start with the H&E or with the PD-L1?
Maybe just a couple of quick things from me, Joe.
Oh yeah,
so
absolutely.
I think it's a correct statement here. So this is from the Pathology Innovation Collaborative Community. So the intention here isn't to advocate for or against anything with the legislation. This isn't meant to be advocacy. This is really just to kick the tires, to work through some of these examples and assess where we think they'd fall in the legislation, what information we can glean from that. And this really stemmed from a practical exercise that Mayo Clinic and a few other academic medical centers went through last year when VALID 2020 came out, where we took a list of, I think, 30 tests, largely, clinical chemistry, microbiology, and that sort of thing. And we independently tried to determine whether which risk classification they would fall into, and therefore what submission would be required. And there was a lot of disagreement. And we found quite often we could sort of talk our way to high risk for just about everything, which is of course concerning. And so as PICC has started ramping up and looking at different projects, Joe and I were having a conversation and thought it would be beneficial to take a few pathology-specific case studies and meet with a larger group and kind of talk through what risk category we think they would fall in, what kind of submission would be required, what elements would be required for that submission, and then finally discussing, would these tests be eligible for the technology certification pathway that's present in VALID. So that's kind of the background of how we got here.
Mm-hmm.
Hey, this is Steve Barbie. I just have a question on the advocacy side. So, what's the current viewpoint of CLMA and CAP and AACC? What's their view of the act?
I don't know that I am in a place to speak to that necessarily. I think a lot of groups are holding their cards pretty close to their vest rather. And I think this group is probably aware there is a competing piece of legislation called the Vital Act, which would explicitly disallow FDA from regulating LDTs and would seek to expand CLIA. So there's a lot of behind-the-scenes negotiating with individual academic medical centers, independent clinical laboratories, and professional societies right now. And so we're trying to stay away from that advocacy piece in this context. Certainly, I'm sure many of us are advocating in other contexts as part of our day jobs, but that won't be the role here.
Okay, thanks.
Yeah, so I think the legislative part is obviously there are many other aspects driving sort of like opinions from the professional societies. I think this is mostly, I think part of what could be the outcome, other than, you know, sharing what we discuss and sort of sharing our opinion or interpretation, would be if truly every single case or every single test case that we're discussing could be somehow argued to become high risk, that the question is how to actually define low risk and or provide some input there. With some of the other projects that we went through similar exercise, we put a little opinion or, you know, a position paper out there, more from what evidence is needed to support this. A prior project, for example, tackled some of the issues about what would this cost, you know, is there additional add-on cost? And one element, you know, if this, you know, holds up as written right now, this would affect software as a medical device as well. That may have huge cost implications. Obviously, there's some intersection with the 21st Century Cures Act and interoperability. So if laboratories build their own software, it may potentially have not direct implications for interoperability, because that may not be, you know, there from the get-go. And then a final statement regarding sort of the overarching aim is the mitigating measures and some of the, let's just call it reference standards or surrogate tools that may result out of this could actually become, at least from a concept, some of the regulatory science tools, right? How would we assess some of these, let's say, PDL1 stain, or what is the ground truth for an H&E stained Gleason grade? So some of it could potentially be done using artificial or surrogate or reference samples. And that sort of interlaboratory variability, which is one of the probably big topics, I think we can discuss, but, you know, I'm not sure whether that is directly solvable just using valid concepts. But so Shannon, maybe you can explain how you did this before to this group regarding the risk assessment that you've done sort of locally, and maybe we can use sort of rinse and repeat and try to tackle it the same way.
Sure, it was pretty straightforward. We essentially had a spreadsheet with the 30 tests. I think we included like the intended use population and maybe a little bit about what the test is used for. And then we disseminated that to the different groups. And these were all pathologists or clinical consultants. And each independently would go through and indicate what risk category they think they would fall in. And then we, you know, kind of gathered together and discussed, well, why did you call this one low risk? Why did you call this one high risk? And it was very informative and a really good discussion. But again, there were many cases. And, you know, I think part of this is, you know, we're laboratorians, and so we tend to automatically go to the worst case scenario, right? And so I think there were many cases where we could talk our way to, well, you know, if the stars align, this test probably could be considered high risk. So I think one of the things that might be beneficial coming out of this as well, to your point, Joe, is considerations or guidance or something like that to help laboratorians maybe not go to that worst case scenario automatically, but to take a more balanced, objective approach when trying to figure out what the risk of their test might be.
I mean, this is just an additional consideration. So Helen had some dealings with the FDA because of COVID. I think many people have. And one of the issues we had with the FDA is the FDA universally jumped to the worst case scenario. So for example, one of the testing strategies we had was a self-collection at home. And so basically we would ship a mid-terminate swab to patients. They would self-collect to themselves and their children, and they ship back. We test for respiratory viruses, including COVID. And we had done this for 2 years, had 18,000 participants. We had one adverse event that was a mild allergic reaction that resolved within 5 minutes. And FDA shut us down immediately upon hearing that we were using a mid-turbinate swab at home unobserved. And the reason is they were worried about every single worst case scenario, impaling yourself. I mean, they literally went through a list that was about 5 minutes long of here's all the possible alternatives. And we said, we have 18,000 data points that say this is not correct. And they said, doesn't matter. So how does that fit into this classification exercise?
Yeah, that's a great observation. Maybe we're not so off base jumping to the worst case conclusion. But I think the concern, and I'm going to try real hard not to get into advocacy, I'll behave myself, Jill, I promise, is, if everything is high risk, then it will bring the clinical laboratory testing industry to its knees. And, academic medical centers that don't have, or that do have a very large, complex test menu just won't be able to support that. So I think it's important that we right size test classification, both for the sake of labs and for the sake of FDA, because otherwise they're going to have to hire 5,000 reviewers, and that's just not going to happen.
So maybe as a quick, I know that many of you are experts in this field, so please stop. But one thing that reminded me of is, of course, the CLIA categorizations, which is the scoring system that is used to delineate test complexity, right? So that's different than risk. But as part of that, obviously, there is, ultimately, I don't know, the scoring amounts to how difficult is it, how complex is it. But I think one element in there is, of course, what is the, what's the knowledge. But all of these components here obviously interfere not with what is the, outcome. So basically an exploratory panel NGS may be extremely complex, but may not put any patient on risk if you just gather data for, epidemiologic purposes or for just data tracking. So I think the argument that you can argue anything into high risk, we can eliminate, because some of the thing is just for data collection. But I think I know what's meant by that. So I'm not sure, is there a definition sort of formally in, other than, putting patients' life at risk, other than what's spelled out in VALID? Because I presume that there must be some ISO definitions or so. Is someone aware?
Oh, almost certainly. There are a lot of different risk categorization schemes. I mean, even if you just look at FDA's medical device framework, they've got basically 3 different classes of risk: low, moderate, high, class 1, class 2, class 3. So that is what has been a little bit mystifying about VALID is the congressional sponsors have been pretty adamant that they want this 2-tiered, low-high paradigm. And then there are a lot of carve-outs as well, like a low-volume tests and humanitarian use and so on. So it's not just those two, but those are the 2 kind of primary.
So what should we use in terms of like a reasonable assumption that, high risk, of course, if someone is being put on the wrong therapy, or if someone is truly being sort of killed?
Well, maybe the place to start would just be to, because I don't know how familiar everyone is with the actual VALID language. So do we want to, and I know Ula put together a fantastic website that includes some of the terminology right out of VALID. Do we want to take a look at the definition of low and high risk just to kind of familiarize ourselves with the language that's used in the legislation?
Sure. Let me just share my screen and then.
So I kind of picked out, 4 terms that I think are relevant for this discussion. First is the actual definition of an in vitro clinical test, which is the new umbrella term for IVDs and LDTs. And notably, it does include under Romanet 4 software. So, for example, AI algorithms used in digital pathology would conceivably fall in here and be considered an in vitro clinical test and be subject to these requirements. So I think that's why the software example is pertinent to this discussion. And then we've got low and high risk, and then finally the concept of mitigating measures. So valid is a little bit funny in that it has kind of a de facto third category, because you've got low risk tests, you've got high risk tests, and you've got high risk tests with mitigations that are not high risk but are also not low risk. So there is sort of a de facto middle road here. That's a hole in legislation right now. So here you see the definition of low risk. So minimal or no harm, minimal or no disability, or immediately reversible harm, and a remote risk of adverse patient impact. That's basically low risk. And then high risk is unreasonable risk for serious or irreversible harm or death, absence, significant delay, or discontinuation of life-supporting treatment. And this kind of gets to what Joe was talking about with the CLIA classification. High risk does account for the intended use of the technology, how well characterized it is. So if this is a brand new technology that no one has ever used before, much greater chance that it's high risk. Whereas if it's a technology that's really, really well understood, so like, I don't know, ICP mass spec or something like that, that would be less likely to be high risk just based upon the technology. Now, if there's still a high risk of, or sorry, if there's still a significant risk of adverse patient effect, then clearly it would still be high risk, but the technology is part of that discussion. And then the part where things get a little bit wonky is B Romanet 1, if there are mitigating measures to prevent, detect, or otherwise mitigate inaccurate results, it's not high risk, but it's also not low risk. So what is it? Good question.
Yeah, I thought that would be a great, example of discussing with the group what you thought of the first case. So let's assume, for the discussion, let's say getting the wrong Gleason grade would be detrimental. Let's just say, I mean, I know it's not quite true because that's not the only determinant, but let's say if that would be wrong, it would be high risk. But you could, as written, it's an AI tool to predict the combined Gleason grade from H&E stain, could, for example, be a mitigation measure to say we take out a certain fraction of cases and have human review it, basically like ongoing surveillance and or, quality control, and we could, discuss whether that could be every case or a subset of cases, and whether such a mitigation or control element within a workflow would classify as a mitigation measure, because the definition of mitigation measures is also very complicated. So what do people think about that?
I think so. Joe, this is Gina. So I think it's really good what you mentioned. I think in addition to that, also, as we know, the intended use is a critical factor, right? So if in the intended use we say assistance for the pathologist or something like that, it depends how that is worded as well. That would help mitigate the risk as well. So if it's an assistant to the pathologist, from my perspective, that would help mitigate things. It just all depends on the intended use, right? That would drive that factor as well.
Yeah, I think, one of the, to just add on to what Gina said, I think that if the test is a sole determinant, like a decision is going to be made based solely on that test, probably going to be high risk, most likely.
Yeah, for sure.
And that's the beauty of pathology in a lot of cases, is many times you're ordering a whole host of stains and tests and so forth to make your decision. But there still could be certain tests that are really definitive that would drive your diagnosis and still could be considered high risk because of that.
I agree, and I'd say that also has regulatory precedent. So, for example, in imaging technologies, if the AI is being used to highlight areas of concern, for example, then it's lower risk. But if it's being used to definitively say if there's a tumor or not or whatever, then it's ranked as high risk. So as long as it's an aid in interpretation or not the sole determining factor, I think there's good regulatory precedent for calling it low risk.
Yeah, I agree
So
for sure.
I agree with that as well. So, but then we could default back to, let's say, I mean, the AI part of this may make it a little bit difficult, but if the test itself, the outcome is high risk, this becomes a moot point, because if you do it with AI, it will certainly be not lower risk. So it must be a low risk test that may become more, let's say, risky through AI, and that gets into the whole discussion of diagnostic aid versus decision support tool versus, no human interaction at all. So the risk then by default, when we look at the AI component alone, defaults back to what is the risk of the original test, and is that increased by AI? But as far as I'm reading this, the question here is more as the diagnostic test as a whole, not as much as the AI part, right? So what I'm trying to say is low risk, high risk in this example then mostly related to the AI component, or would we assume that under valid it would be predominantly what the test is intended to do? Meaning, if the risk for low versus high grade prostate cancer would by default not be a high risk test result, would that have the same implications?
So I do think it comes back to intended use and not necessarily the software, because in the end, AI is an algorithm. It's a fancy algorithm, but it's an algorithm. So the use of software per se should not change the risk of the test. The ability of the physician or whoever, the overseeing person, to intercede or apply their expert knowledge is what would give you the risk determination here.
Yeah, so I think, typically if something is kind of black box, it's going to be higher risk. So if the algorithm essentially spits out and says, your patient has XYZ cancer, and the pathologist is not able to deduce how that conclusion was arrived at and just goes with it, that's going to be high risk. If it's more like the algorithm is, picking out areas of interest, and that drives where the pathologist is looking, and they still use their professional judgment to make a diagnosis, that will likely be lower risk. So I agree. It comes down to intended use and whether a human, speaking about AI now, but whether a human can essentially reconstruct what the AI has determined.
So basically, if a pathologist is able to override, for example, if he or she doesn't agree with the results from the algorithm, then obviously that would lower the risk, right? To your point.
So this is Tom from Euphoria here. So one of the quite often studied effect is that, especially if you have less experienced pathologists, if they first see the AI result, like the highlighted tumor, this is a finding that puts it in high Gleason group, then they are more likely to agree. So they have a risk of overcalling, or they are more likely to agree with the AI result if they see this result first, rather than first making an unbiased determination on their own, and then just using basically AI as a second opinion. So that's kind of the intended use here. Does anybody have any kind of great insight on how the FDA would differentiate between those two, that see the results, then just agree or disagree, or actually first have to do the diagnosis and then see if the AI actually concurs? Because those are two clearly different cases, and there's some studies to say that depending a little bit on how seasoned the pathologist is, they are more likely to agree with a previous point of view.
It's a very interesting point. So I don't have an insight into sort of the ability to, or what the FDA would say to this, but for some of our AI tools that we implemented, we leave 15% of cases out for unbiased review to prevent, when we do relearning, to just relearn on AI calls, right? Which is ultimately creating sort of a snowballing effect. So, but so unbiased review plus, let's say, a decision confirmation versus biased review because you get a decision support or highlighted area. Is that how I'm understanding the question?
Yeah, exactly. Yeah.
So unbiased review plus highlighting versus highlighted initial review or something like that, right?
Yeah.
We can wordsmith this later.
Yeah.
So that's a great point. And so FDA will really look at the totality of all your mitigating measures. So, for example, saying, oh, this is just a diagnostic aid and the pathologist has the final say, that alone may not be sufficient because, for example, you might have a less seasoned pathologist, right? So then you have to talk about, well, here's our training program, or here's our double check, our unbiased review, and so on. So this is a great example, Joe, where again, it's really easy to talk yourself into high risk pretty quickly here because you have all the whataboutisms, right?
One whataboutism is, and I guess that FDA would just ask for data if anybody brought up this point, but it's also very clear that AI, when applied on scale, actually catches misdiagnosis or high grade lesions or some something that obviously is disease that needs to be treated that just was missed, and then quantifying that would require quite a lot of work from somebody, but I think it's very obvious that this does happen. So it's not only that AI actually introduces risk, it actually also in some cases makes better patient outcomes.
Well, this is an interesting question. So what if, for example, the intended use of this first case, to have a whole slide imaging based artificial intelligence application to predict all of this after a pathologist has signed it out, right? That would change the intended use, correct?
Well, if it's after the pathologist has already signed out the case, then it becomes research, right? Like,
it's not diagnostic.
Exactly. So then we're in the clear. It's not a medical device.
Well, unless you amend the case, right? If the human goes back and changes the call. That's why.
I think that would be a very disruptive to the lab flow. Like to have the person fully sign out the case and then reassess it. It would be. I think that would be a very disruptive lab flow.
So just currently in my company, we do that. We do retrospective review of all cases. After they're signed out, we run the AI on it, and then the AI has caught mistakes, and we get another pathologist to look at it. So it's not just the AI result it's just a QC issue, and then the pathologist does amend the case. And so it's currently in process.
Now, obviously, we know that there will never, ever be a computer that's smarter than a pathologist. So let's just put that on the table.
Should I write this down here?
Yes, absolutely.
Quote from Shannon Bennett, 2021.
I have to get my brownie points when I can. So there you go.
That's awesome.
Well, and this idea of retrospective review, I mean, that really kind of fits into what a lot of pathology groups do for QA, right? Where there's certain criteria that are met, and you review those cases as a group, and then come to a conclusion. And so you could say, but again, it comes back to the intended use, right? Like if this is a QA tool versus a diagnostic aid in real time.
Yeah.
And I think as I've walked through Valid and how we would apply it, one of the questions, again, this is really colored by my personal experience with FDA, is basically with everything we start at that high risk, and then we try to work it down to low risk and say, okay, here's the intended use, here's the mitigating measures, et cetera, and see if we can classify it. And and that's really borne out because it's I don't know what the better way to go is. You can either start everything as low risk and try to work your way up, or work your way down. But I think the acknowledgement that, Shannon's little study had, that basically everything could be high risk, is going to be borne out.
So in terms of, let's say for this, the Gleason AI scoring, let's say some of these elements here would hopefully classify as mitigating measures depending on how they're aligned. So basically the one thing that I could hopefully get you to agree on is that the mitigating measures have to clearly be aligned with the intended use, correct? I mean, if that's,
Yeah,
is that
for
what,
sure.
yeah. What about, so go ahead, Shen.
I was just gonna say, on the topic of mitigating measures, there's a little bit of a chicken and egg problem in Valid. So Valid does have a risk assessment, which addresses the mitigating measures, right? So you essentially list out, here are the things that could go wrong. My AI misses a call. I've got a new resident who relies too heavily on the AI, those kind of things. And then you can state, here are the process things we're going to put in place to mitigate those things. And so therefore move it from a high risk to a lower risk. That's great. The problem is the risk assessment is only required for high risk tests. If your test is low risk, you don't need to do a risk assessment. So it kind of reinforces what Eric was saying. Labs are probably going to be best served starting at that high risk, doing your risk assessment, and then figuring out your mitigations. Now that's a tremendous amount of work, but that's probably, that might be the best way to tackle it. Unless something is very clearly low risk where, this test is used in conjunction with other tests, a physician would never act on this test alone. We think this is low risk, that's probably fine. But if there's any question about this test might be greater than low risk, starting with that risk assessment right up front might be the easiest approach.
So what about tech certification for, let's say, this Gleason AI scoring? So let's assume we're in a histology lab, no, and now we're getting a little bit off track, but no higher complexity test, and that would assume that there are higher complexity technologies applied. So let's pick NGS as an example, or whatever, electron microscopy from a complexity perspective. But let's say we're talking about a classic histology plus IHC lab, and this may represent the highest complex technology applied. So I don't know if the word is eligibility or, but whether tech cert could be applied here.
So there's kind of two pieces of tech cert, right? There is your exemplar test that you use to apply for the tech cert. And then there's all the additional tests that you implement after you've received the tech cert. So if your AI is high risk, we determine it is high risk, period. No mitigating measures, it's just high risk. You could use it as an exemplar, but if you have a tech cert for pathology AI or whatever your tech cert would be in, if your test is high risk, it is not eligible for tech cert.
So maybe that was too fast for me. So assume this is AI is high risk.
So there's two steps. There's applying for it.
Correct.
So if our AI is high risk, we can use it as our application IVCT.
Mm-hmm.
And then once we have received our tech cert, hypothetically, we could bring up as many new pathology AIs as we'd like, as long as they are not high risk.
So, but if the initial AI is high risk, we could not bring on any other AI that's high risk?
Correct. If a IVCT is considered high risk, it is not eligible to go to market under tech cert and not be reviewed by FDA.
Okay. So that would mean if the AI initially is considered high risk, we couldn't even apply for TechCert,
You could use it to apply for,
correct?
because then FDA is reviewing that high risk test. Well, let's say we submit our Gleason AI scoring algorithm as our exemplar. FDA gives us a TechCert. Hooray! We now create a second AI software for something else pathology related that is also high risk. We still need to submit it to FDA because it's high risk.
Got it. So in
So
this
Shannon,
case, yep.
oh, sorry,
Go
no. So Shannon, I was curious on your take on this. So my reading of TechCert is that exactly like you said, high risk you can't get TechCert, and low risk you don't necessarily need TechCert. So you don't need TechCert is kind of the end. The only way you can get a TechCert is a high risk test that's been mitigated down to this imaginary medium risk.
Are you implying there's a hole in the legislation, Eric?
I wouldn't say such a thing.
Yeah, exactly. You're right. As the law is written right now, there's basically a tiny sliver of the lab test market that would even be eligible for TechCert. So that's a problem. So I think we could probably run under the assumption that that's going to get addressed, hopefully. And whether that is the creation of a moderate risk category, which is where TechCert becomes very valuable, or if we don't create a formal moderate risk, but we say high risk with mitigations, because those are eligible. So if we have a high risk test, but we're able to mitigate it down to less than high risk, those are eligible for TechCert.
So it could be that TechCert in this case that we just outlined, that an additional AI for, let's say, breast cancer grading with the appropriate mitigation measures would not be initially classified as high risk, but as high risk with mitigation. Therefore, TechCert may be for that particular lab still be attractive to submit the highest risk, right? I think that's the wording in the Valid Act. The highest risk AI tool for the, in this case, H&E and IHC lab. And then, because otherwise, if every single other AI tool is high risk, every single one had to undergo FDA review.
Correct.
So in other words, the TechCert would, however, if I'm understanding this correctly, for additional low risk tests, the lab would basically be covered under TechCert, correct?
Low risk tests don't need to be submitted regardless of TechCert. So if your test is low risk, you don't have to submit at all. So TechCert doesn't come into play. So the only way TechCert will be valuable for a laboratory is if they have a bunch of mitigated high risk tests in a particular technology. So if you have a laboratory that's really, really good at developing mass spec assays, they could get a TechCert for mass spec, and then for 4 years, bring up as many non-high risk mass spec tests they want, bring them to market, and not submit to FDA. That's the benefit of TechCert.
So, but that would be highly attractive for, let's say, areas of expertise. So if we stick to the Gleason AI scoring, so if a lab then decides to do, I don't know, microscopic melanoma invasion or micrometastasis identification, you know, same technology, same, you know, imaging modalities, but different applications.
Mm-hmm.
So if appropriately, let's call it reworded and equipped with appropriate guidance, this new FDA review, as written right now, could become a high risk with appropriate mitigation measures may fall under TechCert.
Correct.
How am I going to write that in 10 words or less? Okay, I try. Keep going.
So maybe you could put, so additional high risk with mitigation, because you've got additional high risk, they're not eligible. But additional high risk with mitigation that fall under that TechCert, the bounds of that TechCert, you could bring those to market without FDA review for the 4-year period of the TechCert.
Is it 4 years? Yeah.
4 years. And then at the end of 4 years, the laboratory would need to send a new exemplar to essentially demonstrate that they still have the expertise in that particular category.
So if then at that time the lab had developed an AI tool to identify tumors of unknown origin without human call, and basically auto-reporting, and that would be classified as the highest risk, right? Then that would undergo tech cert, and then this would be rinse and repeat.
Yes.
That's very helpful. Okay, so other questions about the Gleason AI part here? Doesn't sound like it.
So do we want to, because since we only have 15 minutes, we may not have time to dive into a new one. Do we want to talk a little bit about submission requirements?
Yes.
So, and again, this is kind of a challenge with Valid, where it's kind of an all or nothing. If it's low risk, you require no submission. If it's high risk, you require lots of submissions. So you need to include information about your quality system, quality management system, to demonstrate that the test is being developed and validated in a high quality laboratory, basically, or high quality manufacturing facility. Because again, this is not just LDTs. This is also test kits you buy from your favorite test kit manufacturer.
Mm-hmm.
So quality system information. Of course, you need to include performance characteristics, so precision, accuracy, etc. One challenge with Valid is it is very focused on more of like the clinical pathology versus anatomic or hematopathology, and so that becomes challenging. Like with some of these AI things, how do you translate accuracy, precision, sensitivity, specificity to an AI? It's doable, but it's not an exact fit. So particularly for this use case, there might need to be some interpretation involved. Let's see.
So let me just quickly sort of ask, in this case, right? So what I think you mean is, let's say the Gleason AI score has some sort of probability for, the first or the predominant and then the second part. And I think there are some tools out there that ultimately report out this is a, 3+4 with 98%, probability, or I mean, most of it is probabilistic. And then someone would have to say the ground truth of this is a human call, or a group of human calls, right? Or consensus part. And then we would have, human versus AI, and that would fit into the classic 2x2 square. And I'm not sure whether some of the existing AI tools have been assessed by the FDA as such, or whether they go a little bit deeper. Does anyone know? Is this sort of, is the traditional approach for diagnostics sort of two-tiered, right, wrong? Is that still the most prevalent approach for the classification or for the review?
Well, I mean, this brings up a lot of issues, but and one thing that's really not set out in this framework is this idea of talking with the FDA before you start doing anything, because again, based on personal experience, what we saw was basically we'd submit a data packet based on. So again, this is using EUA, so totally different scenario, but we submit a data packet based on what was laid out in the EUA. And then when they have the review call, they say, oh, by the way, we think that you need to do XYZ on this. And so then we go back and do XYZ, and they give you a week to do it. So you up in the lab for a week, and then you get back to them and they say, oh, that's great, thank you. Now can you go do PDQ? And so, it's one of those things where you need to have a piece of this submission process where you actually go and say, here's what we're proposing, here's what we're going to propose to do, is this good enough, before you even get to submission. Mm-hmm. Just as a adding an additional piece in the scheme.
Yeah, so there is an equivalent to the pre-submission process in Valid, and our experiences were very similar to yours, Eric. So yeah, I think it definitely behooves laboratories to have an initial discussion with the FDA about, here's our experimental design, essentially, our study design. Are we checking all the boxes you expect us to?
But this would be a general requirement, not necessarily to TechCert or undervalid, right? Because there may be different pathways for submissions, correct?
Yeah, that's kind of independent of which pathway you ultimately go with.
Correct.
Other submission requirements, a bibliography. And FDA generally likes a very robust literature review. They really want to know any paper that talks about your device in any way, shape, or form. What we kind of do today is, if we're bringing up a new test, we'll grab a couple of relevant high impact journal articles, and that's our lit review. But that likely will not be sufficient for FDA. They're going to want all literature. So if you're bringing up this AI, you need to do a very deep lit review and bring up any journal article that you can reasonably find that relates to your AI, whether it's supportive of that use or if it is not supportive of that use, and then kind of reconcile that. So that's a significant undertaking right there. You also need to include any raw data that you might have. Let's see, I'm just flipping through. For those of you who want to look at this,
the raw data review is something very interesting. So for some of the AI tools, right, we're talking it depending on what you consider raw data. It may be very beneficial. So for example, for our EUA submission, we sent them like basically our raw CT values and everything, including the curves. Just at that time, that was sort of like, hey, here's everything. You make sense of it as well, right? As we were learning about what this all means. But that raw data for an AI tool for Gleason scoring may be terabytes of data, and may or should probably for clinical use entail personal health identifiers. Right, so this gets into the tricky thing of like interoperability and ability. Even, I presume, on the FDA side, there's a lot of expertise on imaging. I know that, but just data handling, right? So there's a definite pro for saying, hey, look, that's fully transparent, which I think is always good, or with the appropriate skills, that's helpful. But there's definitely also a con on sending some raw data, because that obviously reveals some of the issues. So we had, for example, in one of our commercial reference standards later on, for second submission, we had a plasma contamination that we just couldn't explain. It didn't have anything to do with the performance of the assay, but as a control reaction that we ran, it had basically impact on our control reaction. So that definitely came up, and that delayed some of our approvals for a bit. So.
Then the last two I'd add is a formal risk assessment, which I'd spoken of earlier. And then there are a number of different things that I just kind of lump together as narrative description.
Mm-hmm.
So they want a narrative description of the studies that you're performing. They want a narrative description of alternatives to your test, so that they can assess against those. So this will require a significant amount of what I'll call scientific medical resources, whether that's your lab director or development staff or whatever the case may be. If you are currently developing LDTs, this is going to be a significant change for you.
Yeah, so the one of the biggest advantages of LDTs is that you're not in a you're in a competitive market if you consider that primary aim for the LDT, but I think many folks want to implement a test because they cannot afford, or they want to start with a certain, subset of, let's say, variants, or in this case, probably harness their prostate cancer practice or something like that. So that part here, I think, is a very, very important one, especially for academic or smaller medical centers.
Mm-hmm.
Was it clear from your reading whether under valid, when undergoing this review, that any LDT becomes available for interstate commerce? I mean, this is more a general question.
That's actually a definition within valid. It says that anything offered on patients is considered for interstate commerce, period. It's the same definition they use for tobacco products to bring it under FDA regulation.
So what if you offer an LDT high risk that is not intended for interstate commerce? That would still fall under the IVCT definition, right?
It would.
It's still considered under, it's actually a line in valid that basically anytime you offer a test, it's considered to be introduced into interstate commerce, regardless of whether or not it is.
That's probably the biggest change when it comes to the LDT landscape, right?
Yep.
Other points on our first sort of test drive here. No? So maybe I try to write this down as we discussed this. So the two-tiered risk paradigm, I think Shannon, you had mentioned that that's really the idea to have only 2 tiers. Was this because the moderate group doesn't really apply, or were folks concerned that everything would be placed in the moderate group, or?
I don't know, to be perfectly honest. I don't know why the congressional sponsors have kind of drawn this hard line. There is essentially, as we talked about, a de facto third tier, which is high risk with mitigations. And I know, without getting into advocacy, I do know that a number of groups are advocating for having a formal moderate risk category. So this could change, but with the bill as written, strictly speaking, there are 2 risk classifications with a number of exemptions.
Okay, well, currently this is a 2-tiered risk paradigm that's being proposed, so that should be fine. Okay, well, I will clean this up a little bit, and then we can share this with the via the website, including the video. Maybe we can use the last couple of minutes to figure out a good follow-up time. So I think maybe in 2 weeks. How does that look for everyone? 16th or 16th or the 23rd? 16th? Eric, is that good for you? Norris, Steve, Gina?
The 23rd works better for me, but either one at this time I can probably do.
Either one works for me.
Tom, Norris, Gina?
Yeah, either one works for me as well. Thank you.
Either one works for me as well.
Oh, I lied. I just got booked for the 16th. Sorry. Anyway, schedule the best time that works, and if I can make it, I will. But I definitely appreciate the exercise. So anyway, I have to jump off to my next Zoom. So
perfect.
Take care, everyone.
Let's take the 23rd.
Thanks so much.
Awesome. Thank you so much for joining. Great discussion.
Thank you very much.
Thanks so much.
Thank you.
Okay, bye-bye.